start phylogenetic tree. So we have to understand what is a phylogenetic tree. And about a phylogenetic tree, basically it is an evolutionary tree. You know what is an evolutionary tree or a phylogenetic tree? So here you can find a definition of what is a molecular phylogenetics. It's simply the analysis of hereditary molecular differences. Hereditary molecular differences means molecular means always referring to DNA. DNA, either DNA or RNA. That is a term molecular are always refer to gain information on the organism's evolutionary relationship. For example, we say that human beings and chimpanzee have some connection. If you examine the DNA of human beings and chimpanzee, we can see 98% similarity between human beings and chimpanzee. So that we are proposing that we have some relationship with that animal. But if you examine the DNA of a lower primate, like a monkey, normal monkey, we don't see 98% similarity. So we say that we don't have that much similarity with that species. So according to that, we have now proposed a evolutionary pattern that once upon a time in the evolutionary history, there was a connection between human beings and the apes. That is the relationship that we have proposed. So it is based on this DNA studies. We are now studying that there is some similarity between various types of organisms. So molecular phylogenetics is actually studying that relationship, how organisms are connected, how various organisms have evolved. That is what is done in the molecular phylogeny. So the result of a molecular phylogenetic analysis is expressed in a phylogenetic tree. Here you can find a phylogenetic tree. If you look at that phylogenetic tree or that evolutionary tree, we can use both words. We can very easily understand that these two organisms are more connected than this and this organism. That is the idea that can be that we can easily obtain from by looking at an evolutionary tree. We say that these two organisms, this microsporidia and diplomonadia, they are more closely similar than entamoeba and diplomonadia. So that kind of an assumption we can easily obtain from by looking at a phylogenetic tree or an evolutionary tree. So there is Abin, please don't talk. So going to the phylogenetic tree, this is the main approach in a phylogenetic tree. It is simply an analysis of phylogeny or evolutionary relationship, and that will give you an idea on how organisms are related with one another. Now we are finding what are the techniques and what are the application or uses of phylogenetic tree. You know that every living organism has either a DNA or an RNA, and along with the DNA and RNA, there will be also proteins. Now, one important finding that we have found is that a closely related organisms have higher degree of similarity in their molecular structure. Earlier, I told you that if you take the DNA of human beings and chimpanzee, we are finding 98% similarity because human beings and chimpanzee have closely related. That is why there is higher similarity between human beings and. But if you examine your DNA of human beings and a dog, we cannot find 98% similarity. So we say that there is more dissimilarity. So closely related organisms have higher degree of similarity in their DNA or in their molecular structures, whereas organisms which are distantly related have higher level of dissimilarity. For example, you examine the DNA of human beings and a whale, or human beings and a dog, or human beings and a cat or a bird, we find more dissimilarity than similarity because we are distantly related. But if you find the DNA of human beings and an ape, where we find more similarity rather than dissimilarity. But still there will be some differences, but similarity is more rather and dissimilarity is less. So it is based on this assumption the whole idea of phylogenetic tree construction has been proposed. Another aspect in the here is that conserved sequence. There are certain, for example, when you are studying cytology, there you might have heard about different types of organelles inside a cell. You know what is a nucleus. Along with the nucleus, you have studied mitochondria, endoplasmic reticulum, lysosome, Golgi bodies, all we have studied. And in the mitochondria also, you have studied that there are certain type of DNA. This type of DNA are known as mitochondrial DNA. One important peculiarity or feature of mitochondrial DNA is that they do not undergo evolutionary change quickly. It takes more time for DNA present in a mitochondria to change. That means the scientists are using a term called conserved. Conserved sequence or conserved DNA simply means a DNA that do not change easily. So if you take the DNA inside a nucleus, they can undergo changes more faster than the DNA present in a mitochondria. So scientists are saying that the DNA of a mitochondria are conserved. Simply means they won't change easily or they are more resistant to change. So in evolution, we are mainly taking the DNA of mitochondria because they are not changing fast. They are changing very slowly. So that is why DNA of a mitochondria are usually used in evolutionary or genetic studies. So conserved sequence such as mitochondrial DNA are expected to accumulate mutation over a time. What is the meaning of that sentence? They are expected to accumulate. Whatever changes are taking place in the organism or whatever mutations or evolutionary changes have taken place in the organism, that changes will be reflected or present in the mitochondrial DNA, even though there will not be any morphological change. So if you study the mitochondrial DNA, we can find some kind of evidences that shows that some kind of mutation had taken place earlier in the evolution of this organism. So that is the advantage of studying mitochondrial DNA. Mitochondrial DNA will be carrying some kind of evidences to show that what are the evolutionary changes which have taken place in that organism. So that is the significance of studying mitochondrial DNA. So conserved sequence such as mitochondrial DNA will be carrying the evidences of different mutation that have taken place in that species over a period of time. Assuming that a constant rate of mutation provides a molecular clock for dating divergences. That is, by simply checking the mitochondrial DNA and what are the changes are occurring in that mitochondrial DNA, we will be getting a clear idea of what has happened in the DNA during that time or during the evolution of that organism. So that is the idea of my studying the mitochondrial DNA. It gives you an idea of molecular clock. Molecular clock means if you are examining the evolution of foods, when did the first horse evolved, and after the evolution of the first horse, how many million years have completed before the second stage in the evolution of a horse has started? We will be getting some idea by studying the DNA. So that is the advantage of, that is why recently evolution, genetics and phylogeny are all closely related. So by studying the DNA, we will be getting some idea of how many millions years back this species has changed or this species have evolved. So molecular phylogeny uses such data to build a relationship tree. This is what is known as a relationship tree. Simply an evolutionary tree that shows how how two groups of organisms are related with one another. Then these are the techniques that are used in evolutionary tree construction. Now we will see what is a phylogenetic tree. We have already explained what is a phylogenetic tree. It is a evolutionary tree. It's a tree-like branching diagram. While defining a phylogenetic tree, you can see that it is a tree-like branching diagram showing the inferred. What is the meaning of the word infer? Infer simply means to guess. According to the present evidence, scientists will be guessing that this may be the possible relationship. So, according to the present evidence, scientists are guessing that this is the way the evolution has proceeded. Evolution has gone. So, it is a tree-like branching showing the inferred evolutionary relationship among the various biological species. How various species in the world are related with one another. For example, we are saying that all vertebrates are closely related. These cats, birds, mammals and fishes, amphibians are more closely related. When we are examining the evidence, all have vertebrae. So we place them under one category called vertebrata. So they are more closely related. But if we are taking a vertebrate animal and we are examining with a mollusca or a centipede, we are not finding that much similarity. So we say that they are not closely similar. So it is an evolutionary tree like branching diagram showing the inferred evolutionary relationship among the various biological species. Now, the taxa joined together in a tree are implied to have a descended from a common ancestor. For example, here you can find that this is the evolutionary tree of life, and all these animals, all these species, all these groups are having a common ancestor. So we say that all animals are having a common origin. Here you can see all bacteria are having a common origin. All the plants are having a common origin. So that is how we are proposing the idea. So taxa joined together in the tree are implied. Implied means understood. To have a descended. Descended means formed, evolved from a common ancestor. They have a common origin. That is the meaning. Now, tips of the tree. Now we will come to the phylogenetic tree. I will show you some phylogenetic tree. This is how normally a phylogenetic tree are appearing. Now here, this point is known as the root of the tree. And if you look at this diagram, this is the root of the tree, and this is known the tip of the tree. This is another tip of the tree. This is also another tip. Here also 1, 2, 3 tips are here. Now, what is the sentence mean? The tips of the tree represent groups of descendant taxa. Descendant taxa means what has been formed, what has been evolved, and the nodes of the tree represent the common ancestor of those species. For example, if you look at that, this is the tip and this is the node. So here the taxon A and taxon B are two separate species or maybe two separate genus, and both having a common point here, and this is the node. And node indicates that the taxon A and taxon B have formed or evolved from one common ancestor. That is the meaning. So here this is the tip, taxon A, this is the tip, and this is another tip, which are both represent two separate species. But we have joined both together here, and we have drawn a line below that also. That indicates that there is a common ancestor. That is the meaning. So the nodes here, this is a node. This node indicates that they have a common ancestor. So the nodes on the tree represent the common ancestor of those species or those descendants. And two descendants that split from the same node are called sister groups. What is it? We will have to understand that. Here, this you can see that this is one node, and from that node we can send this is taxon has evolved or formed. From the same node or from the same ancestor, taxon B has also formed. Now, what is the case in the case of taxon C? In the case of taxon C, what we are finding is that is not evolved from this common ancestor. In the case of taxon C, earlier there is another ancestor. For example, this taxon C has evolved from this point. Here there was a common ancestor. From that common ancestor, taxon C has directly formed. Whereas what happened to taxon A and B is that from this common ancestor, another ancestor has evolved. From that ancestor, taxon A and taxon B has formed. Now we are looking, when we are checking that this taxon A, taxon B or taxon C are present in a country at the same time, but their evolution is not same in the same way. Taxon A and taxon B are two separate species. They have a common ancestor, whereas taxon C has another common ancestor. And we say that all the species or all the taxa which has evolved from one common ancestor as sister species. This taxon A and taxon B are named as sister species, whereas taxon C and taxon B are named as non-sister species. So that is just like in a family. So here you can say taxon A and B are sister groups and other are non-sister groups. That is a meaning. And many phylogenies also include an outgroup, a taxon outside the group of the interest. Outgroups stem from the base of the tree, and here you can see that this taxon C is considered as an outgroup. Earlier, I can either call it as an non-sister group or we can call it as an outgroup. Why it is known as an outgroup? There is not much of connection, there is not much similarity between taxon C and taxon A and B. This taxon C has found much earlier than taxon B and taxon A. So that is why taxon C is considered as an outgroup. What is the definition? A taxon outside the group of interest. The outgroup stems from the base of the tree. This is the base of the tree. It formed or stems from the base of the tree. That is why we are calling it as an outgroup. Now there is another term called clade. We have to understand what is the meaning of the word clade. Clade is a group of organisms that includes an ancestor and all the descendants of that ancestor. That is, please read the sentence carefully. It is a group of organism. It's a group of an organ, not one organism. It's a group of organism that includes an ancestor and all the descendants of that ancestor. You can say here that this, for example, this is one species, this is another species. Here there is a common ancestor. Now this represents, this what I am circling is represents a clade. What is the mere definition of the clade? It is a group of organisms that includes an ancestor and all the descendants of that ancestor. What is that? Here there is an ancestor and here the descendants. In Malayalam, descendants means Sandadi Parambaragal. That is the meaning of the word descendants. So, taxon B, here there is a common ancestor and these are the descendants. This circling is representing a clade. Taxon A, B and this common ancestor are together known as a clade. So sometimes when we are dealing with evolutionary papers, you can come across this kind of term clade. Clade simply means a group that consists of an ancestor and all the animals, all the species formed from that ancestor are together known by the name clade. So here you can see clade is an organism that includes an ancestor and all the descendants of that ancestor. Now coming to the applications of phylogenetic tree. This application, so sometimes you can expect a question like that. What are the applications of phylogenetic tree? Several applications are there, several uses are there. Basically, phylogenetic tree are basically used for understanding the evolutionary history, how the organisms are connected with one another. That is the main purpose of any phylogenetic tree, to understand how organisms are related. Then in the case of pathogens or microorganisms, for example, when we are having COVID-19, we say that COVID-19 and the common cold virus are closely related. How scientists have proposed that? Because they examined the DNA or RNA and found that their RNA and the RNA of a cold virus are somewhat similar. So they placed it in the cold virus family or coronidae or coronavirus. That is why it is known as coronavirus. Now from our experience, now we say that that assumption was correct, because the cold virus and the common COVID-19 virus are almost causing similar symptoms. Their way of their way of attacking the body is similar. And after, just like cold virus, now coronavirus is also The symptoms of coronavirus are also gone after one week or 2 weeks. But corona has more higher complication than the common cold. That's the only difference. So it is by studying the DNA that scientists are able to propose the diversity of pathogen, assist in epidemiology of infectious disease. For we have now experiencing that. The scientists are saying that the COVID-19 or coronavirus and the common cold virus are somewhat similar, and their way of spreading is also same. That is why you are putting mask now. And aid in the prediction of function of novel genes. For example, when we have a new gene, we have found a new DNA. We don't know what is the function of the DNA. What we will be doing, we will be taking the DNA and go for the NCBI, and we search. And in NCBI, if we put the sequence of that gene, the NCBI will bring out a series list of search, and that will be showing this gene has 99% similarity with this species. So we can say, if we know the function of that gene, we can easily conclude that this new gene and this has a function similar to this listed gene. So that way we can easily predict the function of a new gene. And also found in biodiversity studies. For example, we are getting a new species. We don't know what is that species. And if you take a DNA from that species and we are doing a NCBA search, and NCBA will show what are the similarities of that new gene with the gene already in the catalog. And from that we can easily find whether this is a new species or this is a species already known. So that way we can this NCA. It is useful in biodiversity studies. Similarly, understanding the microbial ecology, that is also another possibility. For example, we have a new microorganism. When we are checking the DNA of that microorganism, we found that this DNA is similar to the DNA of an organism found in the intestine of a cow. Then we can easily say that this will be also living in the intestine of another organism. So that way we can give an idea of the ecology or life of an organism, to inform multiple alignments, to compare with another DNA, to identify what is the most conserved. Most conserved, conserved simply means occurring without any change. That is the meaning of the word conserved. What is permanent? What is conserved means more or less permanent. They are there without much change. That is the meaning of the word conserved. So these are the steps that will be occurring in the phylogenetic tree analysis. And from this part onwards, it is a little difficult because we are not going to study the complete details. We are only studying a brief idea. And the steps in the phylogenetic tree is shown here. First, we will be selecting an organism. Then we have to identify the molecular marker. Molecular marker simply means a small DNA. A small piece of DNA is known as a molecular marker. Then next step is we are amplifying, sequencing and assembling. In genetics you might have studied, or biotechnology you might have studied. What is gene amplification? Gene amplification means if you have only one copy of gene, we have a PCR like machine, and if you put that PCR machine, it will keep 100 copies or 1000 copies, just like a photostat machine is doing. So that process is known as amplification. Then we will be sequencing the gene, and then we are assembling a phylogenetic alignment, like multiple sequence alignment. Then we are proposing a model. Based on that, we are proposing a model, then phylogenetic analysis there, and finally comes the tree construction. That is the last aspect of phylogenetic analysis. So it's a very complicated process, and only one part that I am taking today. And first part is the basic steps in any phylogenetic analysis is we are assembling and aligning a data set. That is the first step. And the first step is to identify a protein or DNA sequence of interest and assemble a data set consisting of other related sequence. That is how we will be finding out other related species. We will be go to NCBI, and in the NCBI blast we put the DNA sequence. It will be immediately just like a Google search. This NCBA search will be showing you what are the similar sequence. That is the first thing that we will be doing. And once other sequences are collected, we will be doing a multiple sequence alignment. And by doing a multiple sequence alignment, we will be able to identify what are the genes which are similar, what are the species which are similar to this new species. That way, we can be able to find out we can complete the first step, that is assemble and align a data set. Then we have number of programs here. Clustal, WA, MSA, MAFT, T Coffee. These are the names of certain software program that helps us in carrying out this kind of no, multiple sequence alignment. So today we can stop here. It is 11:20 now.